Hello, Brian. This is the effect of temperature on enzyme activity. The role of trypsin is that it's an enzyme found in the small intestines of animals, and it helps facilitate the breakdown of proteins. The question for this experiment was, how do different temperatures affect the enzyme activity of trypsin? Our hypothesis was that as temperatures increase and decrease beyond 37 degrees Celsius, the enzyme will be less productive. The conditions for this experiment was that the enzyme was incubated at either a hotter or cooler temperature to measure the effectiveness at different temperatures and determine whether it was denatured. So our abstract continued. The results were most efficient at 57 degrees Celsius with an absorbance of 1.650 nanometers, and this was significantly different from the other samples according to our t-test. Our hypothesis, however, was not supported. We hypothesized that 37 would be the optimum temperature, but 57 degrees Celsius was actually the most efficient. So continuing on with the introduction, the enzymes are proteins that catalyze chemical reactions by lowering the activation energy. They have a specific active site that binds to the substrate molecule. Enzymes are important because some of the chemical reactions that take place in the body would not be fast enough and would need enzymes to help lower the activation energy so they can happen kind of simultaneously. Trypsin aids in digestion in the small intestine and the pancreas of living organisms. The rationale for our hypothesis I spoke about previously is that in the human body, trypsin is known to be function at 37 degrees Celsius, and therefore we hypothesized that it would function best at this temperature. Moving on to our methods. To start off, we added 50 microliters of trypsin and 450 microliters of reaction buffer into a microcentrifuge tube. From there, we placed the tubes in baths with corresponding temperatures. These were either chilled or heated depending upon the sample. We bring trypsin and reaction buffer up to the temperatures, and after that, 500 microliters of azocasein was added to each microcentrifuge tube, which contained the trypsin and the reaction buffer. These were incubated for 20 minutes in the baths, either the hot or the cold. And after that, they were centrifuged for 2 minutes at 14,000 revolutions per minute. And from there, we collected the supernatant and transferred it into a spectrophotometer tube where we measured the absorbance of each sample. And then we repeated the process again for a second time and third trial and calculated the averages of these. So our methods, the experimental variable was temperature at which each sample was incubated. The control variables included a consistent pH of 8. The time and speed of the centrifuge and the incubation time were all the same. The negative control would be the sample that we did not add trypsin to. And there was no expected change when there was no enzyme added. The substrate is azocasein, which is casein with sulfanilamide randomly attached. It's correlated because the more sulfanilamide in the spectrophotometer tube, the more effective the enzyme is because the enzyme cleaves casein and sulfanilamide through hydrolysis. We use replicates because of it helps show outliers, variability, and accuracy of your results. Experimental observations. After incubation and before centrifuging, the liquid inside the microcentrifuge was orange, and that was expected. After centrifuging once, there was an orange pellet at the bottom of each tube and a yellowish supernatant. The supernatant is the leftover liquid, and the pellet at the bottom was formed because of the centrifuging. After a second centrifuge, there was still an orange pellet at the bottom, but the liquid turned much lighter yellow. These are our results in the table here. We have the temperatures across the top, and across here we have our results. So 57 degrees here shows that it has the highest average absorbance at 1.650 nanometers. Continued over here shows a bar graph, and it shows the standard deviation between our kind of samples, or our trials, I should say. In this one, we have shows the different trials compared to the averages. So this one, we thought there might be an outlier in the 15 degrees because of this first trial that was a lot higher than the other ones. Moving on to the t-test. The t-test is important because it can show you whether or not your results are significantly different or not. So for our 15 degrees, the p-value was not significant. A p-value is significant if it is less than 0.05 or less. So moving on, this p-value for the non-control, the 15, was significantly different. The 25 degrees was significantly different. The non-control is significantly different for this one. The 27 degrees, or 37, sorry, was significantly different. And the 57 degrees was significantly different only for the non-control. And for the last one, 70 degrees, the non-control was not significantly different. The experimental design was adequate to test our hypothesis. The measured data had mostly consistent results. There was difficulty in staggering the times because of just human error. And, the results indicated that the trials for the experimental group 57 degrees had the highest enzyme activity on average, and the hypothesis is therefore not supported because of this. I think that's it.